Hello everybody and welcome to our BrainVision Analyzer 2 webinar. As implied in the topic, it's going to be an introductory webinar on source analysis. You are about to grasp the basics of source analysis, mainly focusing on distributed source imaging using LORETA. Let me first introduce ourselves to you. My name is Kedis Mideksa and I will be your today's presenter. With me is my colleague Michelle Hofstetter as moderator. So in the next 90 minutes or so, the intended goals are first to make you familiarize with the rationale behind source analysis by diving into its basic theoretical background. Next, we will focus mainly on LORETA, which is a well-established and widely used source imaging algorithm in hundreds of scientific publications. And of course, this algorithm, which has gained such a widespread popularity, is also what's implemented in Analyzer 2. Hopefully, by the end of the webinar, you would be highly motivated and encouraged to make use of LORETA's functionality as part of your processing pipeline and broaden your research questions beyond the scalp domain analysis by diving into the source domain analysis. Moreover, you will also learn how to extract some relevant information from these source domain analysis. Okay, let's move on to the contents for today's session. We will begin with the theoretical background of source analysis, including the rationale behind it, along with the two main ingredients of source analysis, which are the forward and inverse problems. In the second section, we will focus on LORETA and talk about its theoretical basis. Then the third section will be about getting to know its implementation in Analyzer, including its limitations, and also create awareness of certain rules that should and should not be done before applying LORETA in Analyzer. In this section, we will also open up Analyzer and demonstrate how to use LORETA and also how to extract certain measures from the output of LORETA using a simulated and experimental data. Then we will have time to address some questions of interest which we have collected during the live webinar. Finally, we will close the webinar by providing you the main take-home message as well as some resources where you can learn more about LORETA and general source imaging. So let's go step by step and start with some theoretical background. To start with, this webinar is about EEG source analysis. But as you know, there are different kinds of neuroimaging modalities apart from EEG. So let's ask ourselves, why are people so fascinated about different modalities? Well, there is no secret about it. It is the need of achieving better resolution. There are at least two kinds of resolution. One is the spatial resolution, which tells you how small or how detailed you can see things happening inside the brain. And the other is the temporal resolution, which tells you how fast you can capture changes inside the brain. There is not yet a single modality that delivers a high spatial and temporal resolution at the same time. The existing modalities are grouped into non-invasive and invasive measurement techniques. The invasive approaches include electrocorticography or ECOG, which involves implantation of electrodes into the tissue of the brain, which when stimulated gives rise to a local field potential. With the invasive approach, one can even measure spikes directly arising from single neurons, thus providing a relatively high spatial resolution. Although the invasive measures provide high spatial resolution, they come up with a risk of brain infection and tissue damage during the surgery for electrode implantation. On the contrary, non-invasive measures have become a valuable tool as they provide direct and indirect measures of brain activity without the need for surgery. Functional magnetic resonance imaging, or fMRI, is one of the common indirect non-invasive brain imaging approach. It provides an excellent spatial resolution but poor temporal resolution, as changes in blood flow lag a couple of seconds after change in neural activity. Among the direct non-invasive approaches are electroencephalography or EEG, which records electrical field at a distance using electrodes attached to the scalp, and magnetoencephalography or MEG, which records the magnetic field at a distance from sensors distributed around the head. They provide an excellent temporal resolution and detect changes within a millisecond time frame. On the contrary, they have a coarse spatial resolution. The in-between modality that provides suboptimal spatial and temporal resolution is functional near-infrared spectroscopy, or FNIRS. However, it is not a direct measure of neural activity, as it measures changes in cerebral blood flow. So each method has its own strengths and weaknesses, and selecting a single method or combination of methods really depends on the experimental conditions. One common approach to achieve high spatial and temporal resolution is to combine fMRI with EEG. But what makes EEG still an attractive modality for the functional study of the brain? Let's see by comparing it with that of the fMRI. EEG provides an excellent temporal resolution by recording brain activity on the order of milliseconds, as opposed to fMRI. The spatial resolution, on the contrary, is rather poor as opposed to fMRI, because the electrical activity is summed over space and the signals also get distorted until they reach the scalp while passing through the tissues of the brain. EEG measures a direct consequence of the electrical activity of the brain as opposed to fMRI, measuring the metabolic activity, which is a non-direct measure of neural activity. Although they seem to complement each other, they measure different things, that is, electrical activity versus metabolic changes. Unlike fMRI, EEG is portable, and it has an affordable lifetime price, which includes both the cost of initial purchase and the cost of maintenance. So this leaves EEG to be an excellent modality that can also be used in real-time operations, having a superior temporal resolution with the cost of inferior spatial resolution. Even with this cost of poor spatial resolution, it is nevertheless possible to get a general idea about where the activation might be occurring with the help of appropriate source analysis techniques. So this leads us to the main aim and applications of source analysis. To start with, you can think of the brain like a committee of experts. It is organized into different parts that are wired together in a specific way where each part has its own special task. Once you have measured the summation of the electrical activity coming from the different parts of the brain using EEG, you have been using several signal processing techniques at the scalp level. Unfortunately, the scalp level analysis faces a problem in a way that the signals measured on the scalp don't necessarily indicate the active neurons in the brain. So you might be interested of applying source analysis that enables you to trace brain circuits and unlock the secrets of the brain. So the aim is to find the underlying sources of the scalp recordings by analyzing the brain activity using the technique called source analysis. It helps you to map brain regions responsible for a specific function and behavior. For example, right now, if I stop talking, the areas at the back of your brain called the occipital lobe, and specifically your visual cortex, will be highly activated while you are reading the sentence in the slide. Some applications in which source analysis has contributed to scientific and medical research include the state of eyes closed at rest, generating the alpha waves, which has been shown to be dominant over the posterior regions. Another experimental paradigm is the auditory evoked potentials arising from acoustic stimulation. Their source generators have been shown to be dominant over the temporal lobe near the auditory cortex. Another evoked potential paradigm is the somatosensory evoked potential, which is generated in response to electrical stimulation of peripheral nerves. The source generators have been shown to be dominant in the posterior bank of the central sulcus, contralateral to the stimulated nerve, as the brain is cross-wired with the left hemisphere controlling movement on the right side of the body, and vice versa. Source analysis techniques have also been developed to provide insights on cognitive processing, which is the core function of the frontal cortex. It has also been shown to be a very useful tool in clinical environments as a pre-surgical planning for patients diagnosed with different forms of epileptic spikes, or seizures in a non-invasive way by localizing the epileptogenic zone responsible for creating the abnormal EEG signals. So, what is the rationale behind source analysis? EEG provides the relative measure of the brain activity non-invasively. This means we get reference-dependent scalp measurements as a voltage difference between two different recording sites, with one site serving as a reference for the other. Since it's a non-invasive measure, we can't get the activity of a single neuron. Rather, what we get is a superposition of different source activities. For instance, if we have a source at the back of the brain, and if we have simultaneously active source at the front of the brain, the channel close to the back and the channel close to the front source will primarily pick up the respective sources, whereas channels in between will pick up the activity from both sources. Especially, both sources project to all the electrodes. So the consequence for the measured scalp data is that it will be the superposition of time series of different sources. So source analysis is the approach that mitigates the above-mentioned limitations of EEG by achieving a reference-free data representation, by decomposing the overlapping EEG patterns into its underlying sources and localizing them within the brain. Source analysis is not only answering the question of where the sources are, it also provides you with the time courses of each source. So, what are the ingredients of source analysis? The first ingredient is forward modeling, which involves calculation of the electrical potentials generated from the neuronal current sources propagating through a volume conductor medium. This volume conductor is basically a region or volume of the tissues inside the head. Each tissue of the head is electrically conductive and it acts as a transporter of electromagnetic fields from the neuronal current sources towards the measuring electrodes. So, determination of this field is what is known as the forward problem. And the second ingredient is inverse modeling. That means if we proceed in the opposite direction and try to reconstruct the underlying neuronal current sources from the measured electrical potentials. So, finding out the location and magnitude of these red blobs or activations inside the brain is what is known as the inverse problem. So let's start with forward modeling. It basically comprises of the following four ingredients. Of course, what might be interesting is to go directly to the inverse modeling and find out how to reconstruct the sources. But we can't solve the inverse problem without solving the forward problem. So let's dig in briefly into each of the ingredients step by step. So the first ingredient is source modeling, that is, how to model the neuronal current sources. In order to talk about source modeling, first let's talk about where these sources are originated from. Remember that EEG oscillations arise from the interplay of many neural ensembles through excitatory and inhibitory synapses. What you see here on the very left is the zoomed version of a single pyramidal cell. These pyramidal cells are usually found spatially aligned in the most superficial layers of the brain, and their activity is synchronous, which provides the largest signal that can be measured from the scalp. These electrical signals transmitted along a dendrite creates charge which acts as a battery, known as primary currents, with a positive polarity where the current is leaving and a negative polarity where the current is returning. This separation of charges is similar to a dipole. So sources representing synchronously active pyramidal cells can be modeled using an equivalent current dipole. A dipole is characterized by its 6 parameters, 3 for its location and another 3 parameters for its vectoral components, that is, the x, y, and z components of the dipole in the Cartesian coordinates. These vectoral components can also be transformed into spherical coordinates, resulting in its magnitude and its 2 orientations, that is, an azimuth angle phi and an elevation angle theta. So we have seen that the source can be modeled using a single dipole. It is the simplest model used to model neural ensembles, located in one side as shown here with the red circle. All the small red arrows can be modeled with a single larger equivalent dipole. Mathematically, a single source is expressed using a 3x1 column vector representing the direction and magnitude for each of the x, y, and z components. The advantage of this single dipole model is if you have a very focal source or point source. For example, basic sensory responses, like auditory responses, are known to have focal sources that are far apart from each other on the two hemispheres, and they can be modeled separately using a single equivalent dipole. However, most of the sources, like those of generalized epileptic seizures, are not point sources, but they are quite extended. So these kind of sources give rise to a multiple or distributed dipole model. It enables to model sources across a small cortical patch or all over the cortical sheet. Likewise, the mathematical expression can be extended for multiple sources, and it is denoted by a matrix J with a dimension of 3m by 1, 3 representing the x, y, and z components, which are represented by the arrow vector J, and m representing the number of sources. Okay, let's move on to the anatomical and head modeling, which are the second and third ingredients of forward modeling needed to determine the volume conductor. So first, the anatomical modeling. An MRI scan is commonly used to acquire anatomical images. However, there are certain differences, for example, in the brain's volume, shape, and size across different people. Due to this reason, there comes a need for a reference or a brain template, which is also known as a standard brain template. They represent finer anatomical details of the brain. Brain templates can be generated from a single representative subject, where Talaric and Tourneau constructed the brain atlas from a single post-mortem brain of a 60-year-old French woman. This atlas has become one of the standard reference systems in human brain mapping and enables to identify and label anatomical details of the brain. The more advanced brain templates are generated using the average of multiple subjects' MR images. These are the most commonly used adult brain templates presented in chronological order. These templates have higher signal-to-noise ratio and provide better contrast between the gray and white matter. The MNI 305 and MNI 152 brain templates, created by the Montreal Neurological Institute, are generated by averaging 305 and 152 brain MR images of normal subjects. The ICBM 452 brain templates, created by the International Consortium for Brain Mapping, are generated by averaging 452 brain MR images of normal subjects. The Korean, French, and Chinese templates are created by the respective community to emphasize the need for population-specific templates. So, having a standard brain template helps you for a group analysis, that is, to quantify typical variations within the study cohort. Moreover, it helps you in presenting your results in a well-known template space, the common ones being the MNI or Talaric space. However, if you are interested to perform subject-specific study, an individual brain is of desire. That means to use the T1 weighted MR image of that particular subject for building detailed models of subject-specific head and tissue geometry. So this leads us then to the head modeling. The first volume conductor model of the human head consisted of a single-layer spherical shell. Since the head was assumed to be a homogeneous, isotropic, and resistive medium of spherical shape, this model also assumes to have a uniform conductivity throughout the head. However, it was noticed later on that the skull tissue has a conductivity which was significantly lower than the conductivity of the skull and brain tissues. So this gives rise to the 3 or 4 shell concentric spherical models. Here on the right, you can see the four-shell spherical model, where the innermost sphere represents the brain, followed by a second sphere representing the cerebral spinal fluid, or the CSF. The intermediate sphere represents the skull, and the outermost sphere represents the scalp. Each layer has a homogeneous isotropic conductivity. All spheres share the same center with different radii. This radii can be adjusted to fit a sphere to the MNI brain. Because of their simplicity and ease of computation, the three-layer spherical models are routinely used in most clinical and research applications for approximating the brain, the skull, and the scalp. On the other hand, since our brain has an arbitrary non-spherical shape, it gives rise to the second type of head modeling. So the second type of volume conductor is the realistic head model, used to achieve an accurate approximation of the entire brain. For this purpose, the subject-specific MR image is first segmented to separate the MR image into the different tissues. For example, into 4 tissues consisting of the brain, CSF, skull, and scalp. And then their model is constructed. One of the simplified realistic model is known as the boundary element model. In this model, the tissues between the boundaries are assumed to have homogeneous and isotropic conductivities. One can still claim that our head has anisotropic conductivities. To address this issue, there are 2 computationally expensive models called the finite element method and the finite difference method, which considers anisotropy within a tissue. That means it assigns different conductivity values inside each triangular mesh. The major difference between the boundary and the finite methods is on how they describe the geometry. In the boundary element method, the geometry is described by boundaries using small triangles, whereas the finite models usually work with triangular pyramid, also known as tetrahedrons. So this is just to briefly mention the existing realistic head models and increase your appetite for further reading. We will not go in depth into this topic. So for those of you who are interested to know more about the pros and cons of these methods, you can also have a look at this journal article entitled Effects of Forward Model Error on EEG Source Localization. It was authored by Zainab Ajash and Scott McCaig. Now let's move on to the last ingredient of forward problem, which is co-registration, also known as coordinate transformation. All the models that we have discussed, that is, the anatomical and head model, as well as the electrode positions, must all be expressed in the same coordinate system in order to compute the electrical signals produced by a dipole. So as I have mentioned earlier, the Thalarac atlas with its coordinate system is used as a reference, and the anatomical model, that is the MNI template, is registered to this reference atlas in such a way that it matches the equivalent positions on the Thalarac atlas. Then this registered MNI template, which then forms the 3-dimensional standard MNI brain, is used as a reference for the spherical head model to be registered to. Finally, the electrode locations are projected on the co-registered head model. So most commonly used external fiducial markers or internal anatomical landmarks to define the coordinate system are the Nazian points, describing the y-axis, which is the posterior-anterior direction, and the right and left preauricular points, describing the x-axis. The intersection of these 2 axes makes the origin, and the z-axis basically points towards the tip of the head, forming right angles between the x and y-axis. So far we have discussed how to model the neuronal current source and the tissue of the head or the volume conductor model. Now let's move on to the final stage, which is solving the forward problem, that is, calculating the electrical potentials. So given the source model parameters, that is the position, orientation, and magnitude of any neuronal current sources, along with the geometry of the head, that is the volume conductor model, what is wanted here is to calculate the voltage distribution over the scalp. So a mapping function or a projector is needed that shows the linear relationship between the neuronal current sources and the voltage measurements. This mapping function is known as the lead field matrix or the gain matrix. Mathematically, for n number of channels and a single source, it is represented by a matrix G with a dimension of n by 3. 3 representing the gain vector for the x, y, and z components, which are denoted by the matrix small g. Similarly, the matrix can be extended for m number of sources having a dimension of n by 3m, where the gain vector for each of the 3 components are denoted by the matrix small g. So, solving the forward problem, as I have mentioned before, is determining the scalp topography or the voltage distribution from the neuronal current sources modeled as dipoles. So, the physics of going from the source to the voltage is governed by the quasi-static approximation of the Maxwell's equation. Due to this approximation, it leads to the simplified equation known as the Poisson's equation. Sigma is the conductivity of the volume, and nabla indicates the derivative function. So the multiplication of the conductivity with the spatial derivative of the electrical potential gives the current source density, and taking its derivative gives the volume current density. This equation depicts the linear relation between the electric field and the volume current density. This linear relationship is mediated by the lead field matrix, which contains the conductivities of the tissue and the geometry of the head. So the electrical potentials are calculated as a multiplication of the lead field G and the source J. Okay, I will now summarize briefly the whole story of forward modeling. EEG signal is generated by neuronal current sources, and these collections of current sources are modeled using equivalent current dipoles. The bridge between the source and voltage matrix is mediated using the lead field matrix, which consists of certain source parameters describing the physics of how currents behave inside the head. It also consists of the volume conduction model, providing information about the geometry of the head and the conductive properties of the tissue. So, what did you benefit from solving the forward problem? It enables you to calculate the potentials generated by the neuronal currents, which makes it a necessary prerequisite for solving the inverse problem. So we have covered the first ingredient of source analysis, which was forward modeling, and now I will move on to the second ingredient of source analysis, which is inverse modeling, that is, reconstructing the neuronal current sources. To start with, you have the voltage measurement that you obtained by solving the forward problem. And what is wanted now is to estimate the neuronal current sources described by their positions and orientations in the brain, along with their magnitude variation in time. So as it was in the forward problem, here also there exists a so-called generalized inverse matrix that relates the voltage to that of the source. So the multiplication of this matrix with the voltage matrix gives you the estimated current source matrix J hat. The inverse problem is very challenging to solve. This challenge can be better illustrated by this classic example. It is the problem of a knight trying to guess the species of a dragon. He is trying to hunt a particular species just by observing the footprints he finds in the forest. Well, this is a very difficult task if many species of dragon produce similar footprints, and it becomes even impossible if the footprints are slightly smeared in mud. So, for the knight, determining the footprint, having an a priori information about a particular species of a dragon is pretty easy, and it gives him a unique solution, which is an analogy for the forward problem. However, inferring the species of a dragon from infinite number of footprints will be a nightmare for the knight, which is the analogy for the inverse problem. So the inverse problem is known to be an ill-posed problem. Ill-posed problems basically violate one or more of the criteria of well-posed problems. So when do we say a problem is well-posed? The problem must have a solution. The solution must be unique and stable under small changes to the data. So when these three criteria are fulfilled, then we say the problem is well-posed. If we relate this to our brain inverse problem, there are infinitely many source configurations that can give rise to the same voltage distributions over the scalp, making the solution to have non-unique solutions. That means there are infinitely large number of sources M as compared to the number of channels N. The other point is that the estimated sources are sensitive to noise, making the solution to be unstable, violating the third criterion of well-posed problem. So, how should you handle the ill-posed nature of the inverse problem? You can resolve this ambiguity by setting certain a priori assumptions or constraints which help in constraining the space of the feasible solutions. One of the constraints is known as functional constraints, where you can set the number of dipoles a priori, or where you can either fix the location or the orientation of the dipoles, or both, which leads to a smaller number of parameters to be estimated. For example, unconstrained dipoles, also known as moving dipoles, which are free to move and rotate, leads in the estimation of all the 6 parameters, 3 for the location, 2 for orientation, and 1 for magnitude. However, if you assume your dipole to be freely rotating, but restrict its location to be fixed, this leads to the estimation of only 3 parameters, 2 for the orientation and 1 for the magnitude. If you even restrict the location and orientation of your dipole model to be fixed, then only the magnitude needs to be estimated. So, functional constraints are constraints applied on the dipole model. The other constraint is known as mathematical constraints, where you can set constraints on the mathematical equations. For example, minimum norm is one of the mathematical constraints that minimize the differences between the true source and the estimated source, and it searches for the solution with a minimum power. The search for the solution with a minimum power from all the possible solutions is a constraint that warrants a unique solution. Another example is maximum smoothness that can be imposed as an additional constraint to the source distribution that expresses smoothness in the source distribution. So, searching for the maximum solution from all the possible solutions is another constraint that warrants a unique solution. The other family of constraints is known as anatomical constraints, where you can set constraints on the source space. This means you assume sources to be located within a grid of a desired cortical space, where each grid can consist of a single dipole with its 3 components. Just to add a word of caution here, when combining prior knowledge with that of the voltage data, care must be taken not to give too much emphasis on the prior knowledge, as the inferred solution will correspond exactly as expected. For example, when you prepare a chicken soup, you add salt to bring out the flavor in the broth, just as prior knowledge should be used to enhance the information content of the data. But adding too much salt, however, results your soup to taste like salt, destroying the flavor. So there should be a balance between the model data and the real data. There are various number of inverse methods. The first category is known as parametric approach. It is based on single and multiple dipole models. Dipole fit is one of the very first inverse modeling algorithm that is grouped under the category of parametric approach. The main assumption here is that the number of sources must be known a priori. This approach enables you to estimate the location of the strongest source. The second category is known as non-parametric approach. It is based on distributed source models. Minimum norm, LORETA, S-LORETA, LORETA, and SWARM are among some of the algorithms that are grouped under the category of non-parametric approach. Unlike the parametric approach, here you don't need a priori information about the number of sources, but rather you determine the location of the source space. This approach enables you to estimate the distribution of source activity throughout the predefined source space. The third category is spatial filtering. One of the spatial scanning algorithms is the music algorithm. The other class of spatial filtering methods is beamforming, which comes in different approaches depending on which domain the estimates are performed. The LCMV and SAM rely on time domain estimates, whereas DICS rely on frequency domain estimates. The main assumption here is that sources should be temporally uncorrelated. This approach enables you to estimate the contribution of a source to the measured data at a specified location, while avoiding the crosstalk from other regions. So, why do we have all these methods? It is because there is no gold standard method available yet. The relative strengths of different inverse methods offer an opportunity to select the most appropriate algorithm for a given experiment or research paradigm. Thus, LORETA, which is one of the non-parametric approaches and which stands for Low Resolution Brain Electromagnetic Tomography, is a well-established and widely used source imaging algorithm in hundreds of scientific publications. And of course, this algorithm, which has gained such a widespread popularity, is also what is implemented in Analyzer. So let's first dig into its theoretical basis. First, what is LORETA? It is one of the most established current density reconstruction methods. It enables you to estimate the distributed activity throughout the brain volume. So first you start with defining a source space inside the brain volume. The most commonly used source space is the cortex. Then this cortical space is discretized into a dense 3D grid, and a single dipole is assigned on each of the grid points. Conceptually, it is assumed that these dipoles are fixed in position within the predefined source space. So the goal is then to estimate the magnitude of these dipoles at each grid point. These grid points are also referred as voxels. There will be infinite number of dipolar sources that can give rise to the same voltage distributions over the scalp. So as I have mentioned earlier, additional constraints are needed to obtain a unique solution. It is assumed that the activity from neighboring sources tend to be correlated or synchronized. That is, they have similar orientation and magnitude across space. Based on this assumption, the mathematical constraint that is imposed on LORETA is that it should select a solution with the smoothest source intensity values. Mathematically, the estimation of the source strength is based on the least squares technique, which attempts to find the optimal source strength J that minimizes the square of the differences between the measurement V and the model G times J. Since there are infinite number of J's that can minimize the data term, an additional term known as the source term is added to compensate the non-uniqueness. This source term basically imposes constraints to the sources, that is, the source J is multiplied by a depth weighting factor W in a way where larger weights are assigned to deep sources to better localize them. Moreover, these weighted sources are multiplied by a discrete Laplacian operator B that expresses the smoothness in the source distribution. Both the data term and the source term are linked with the regularization parameter lambda, which balances the data term and the source term. So this constraint warrants a unique inverse solution. And this unique source strength that minimizes the two terms and at the same time provides the smoothest possible intensity values is derived by the matrix multiplication of the generalized inverse matrix T and the voltage matrix V. T is obtained by the following matrix manipulations, which involves the depth weighting matrix W, the Laplacian operator B, and the lead field matrix G. So if you have two distinct sources, LORETA basically provides a blurred, that is an over-smoothed source distribution due to the assumption that the activity of neurons in neighboring cortical patches are correlated. So the characteristics feature of LORETA, as its name also implies, is that it produces a low-resolution tomography. The nice thing about it is that even if it produces low resolution, it preserves the location of the maximal activity. Okay, I will now summarize briefly the whole story of source analysis. We have two domains. One is the electrode space, where we have the voltage measurements, and the other domain is the source space, where the neuronal current sources are confined inside the brain. These two domains must be co-registered so that both will be in the same coordinate system. The forward problem takes information about the electrode positions from the electrode space, and it also takes information about the source parameters from the source space, along with the volume conductor geometry, which is stored in the lead field matrix G. The forward problem is then solved by multiplying this matrix with the true source J, providing the voltage values. Next, the inverse problem takes information about the true voltage measurements from the electrode space, as well as the calculated voltage from the forward solution. The problem is then solved by multiplying the generalized inverse matrix T with the calculated voltage, providing the estimated sources J hat. This problem is ill-posed, as I have explained earlier. So, additional constraints are needed to warrant a unique solution. For example, if you see this image, you might think that it is a shadow of a wolf, or you might even think that it's a shadow of Spack demonstrating the live long and prosper finger thingy. It's hard to tell, right? However, if I provide you with a priori information, whether it's a person or an animal, then your guess will reduce, and you will get close to finding whose shadow it is. So be aware of the necessity of the constraints that has to be set to achieve a source that is very close to the true source. Okay, now I will move on to the implementation of LORETA in Analyzer and its ingredients. So as I have already mentioned in the introduction section, the first ingredient is source modeling, where sources are modeled using equivalent current dipoles. So how is it done in Analyzer? First of all, a source space is defined which is restricted to the regions of the cortical gray matter and hippocampus in the thalaric atlas. These regions are determined by the corresponding digitized probability atlas of the MNI. So the model takes into account those sources that are confined in this predefined source space. By now, you might have already recalled what this constraint implies. It is the anatomical constraint. So assuming this anatomical constraint, the source space is discretized into a total of 2,394 voxels at 7 mm spatial resolution. These are the three different planes to help you visualize how the grid points or the voxels are distributed throughout the source space, covering those gray matter areas belonging to the cortical and hippocampus region. The axial or the transverse plane is when observing the source space from top to bottom. The lateral or the sagittal plane is when observing from side to side. And the coronal plane is when observed from front to back. So as you might have already expected, the next ingredient is the volume conductor model described by the anatomical and head modeling. So in Analyzer, we offer standard brain MNI templates, which is based on the MNI 305 brain template. For the display purpose, that is to enable you to visualize the mapping of the source activation to different brain anatomies, we also offer a high-resolution standard brain template acquired from one of the MNI lab member, Colin Holmes. So you have the possibility to select the images weighted either by the proton density, or by T2 or weighted by T1. The MNI images are co-registered to the thalaric brain atlas to establish the detailed brain structure in the MNI space. So for the standard MNI brain, we offer a 3-shell spherical head model, which is fitted to the MNI brain template, which has been co-registered to the thalaric brain atlas. The last step is to co-register the electrode coordinate to that of the head anatomy. In Analyzer, electrode coordinates are expressed in spherical coordinates. That is, the position of an electrode is specified by its radius r, elevation angle theta, and azimuth angle phi. In Analyzer, we use the right-hand rule convention for representing the x, y, and z planes. The positive x-axis points towards the right ear. The positive y-axis points towards the nose. And the positive z-axis points towards the tip of the head, and the intersection of these lines is our origin. Considering the standard 1020 electrode configuration, the x-axis passes exactly through T7 and T8 electrodes. The y-axis passes through OZ and FPZ electrodes, and the z-axis runs towards the CZ electrode. Since we have a spherical head model, the radius r will always be 1 for all scalp electrodes and 0 for those non-scalp electrodes. An example would be the ECG electrode. All of these ingredients, that is the source modeling, anatomical modeling, and co-registration, are already inbuilt in Analyzer, and you don't have to perform any of the steps manually. It is just to have an impression of what is implemented in Analyzer, and most importantly, to have a better understanding, as you would need to describe them while writing your scientific manuscript for publication as part of the relevant parameters for your source analysis results. Okay, now it's almost time to go to Analyzer. But before that, let me briefly introduce you to the datasets. For the demonstration, I will be showing you two different datasets. One is a simulated data, and the other one is an experimental data. For the simulated dataset, two sources were simulated. The first source is simulated by placing a radial dipole in the occipital area, which is perpendicular to the scalp, and it was sitting at -3 in the x direction, -72 in the y direction, and 1 in the z direction of the MNI coordinate system. And the second source is simulated by placing a tangential dipole in the frontal area, which is parallel to the scalp, and it was sitting at -3, 52, and 1 for the x, y, and z directions. Both sources have their own independent waveform of similar amplitude, and they were separated by 90 milliseconds. So now let me start with this simulated data and demonstrate the LORETA module in Analyzer that combines the forward and inverse solver in one go. Okay, let me open Analyzer. And then I will open the simulated history file. So, as you can see here, the resulting voltage distribution, as it would have been recorded as a consequence of the simulated brain activity, is depicted using 64 electrodes, and an additional CZ electrode was used as a reference channel. And the data was sampled at a sampling rate of 200 Hz. And as you can see here in the navigation bar, 100 of these simulated waveforms were used to simulate an evoked response. So this continuous data was then segmented based on these triggers, and then it was averaged. So to clearly visualize the data, I have prepared a montage with the selected electrode. Let me open that montage. So you see I have prepared already 3 electrodes. One is from the front, which is close to the frontal source, and one is an occipital electrode that is very close to the occipital source, and the third one is from the frontocentral area, which is a bit further from both sources. So as you can see here, the frontal electrode and the occipital electrode have picked up their corresponding sources. But if you see at the middle electrode, it has picked up the superposition of both signals, and this is due to the volume conduction effect. So now let's try to disentangle this mixture of sources using LORETA. So let me go back to the standard montage and work on the average node. For better visualization, I will switch the view to a butterfly view mode. Let me increase the amplitude a bit. Okay, so in Analyzer, there are two ways to access LORETA. The first option is to access it via the transient mode. So to do that, you need to select an interval like this using your mouse, and automatically when you release your mouse, a context menu will appear, which lets you to select LORETA. Okay, so if you click on it, a dockable extra window will be generated as you see here, and it contains the estimated source activation for the selected data points. So if you see here at the status bar, it shows you the time interval which you have selected in seconds, and then this number inside the parentheses indicates the coordinates of the voxels where these yellow sliders are positioned, and this next number indicates the current density value in microampere per millimeter squared. And this is basically the magnitude of all the current density vectors within this selected time interval. So if you now hover around the activated region, you will get the corresponding current density magnitude for the voxel coordinate where this crosshair is pointing to. So you see when I'm moving, the current density value is being adapted on the fly. So to make sure that the maximum current density is displayed, what you need to do is you can right-click inside this dialog and then make sure that the option, this log navigation to maximum, is selected. So by default, this option is already selected. So what you see here at the bottom in the status bar is the maximum current density magnitude for the selected time interval. And the yellow sliders are already pointing to the strongest activation. So now I will drag this selection bar to see the change in the source activation along time. So over this time interval, the occipital source is still dominating. So that is why you see the activation only in the occipital area. But now, when I reach the time interval where the two sources superimpose, you can see that the frontal source starts also slowly appearing. You see, and then when I'm about to leave, where the contribution of the occipital source is not anymore there, so the occipital source dies out, and then the frontal source dominates. Okay, so this way you can inspect your EEG data. So LORETA as a transient mode enables you to inspect your EEG data and have a first impression about the location of the activated sources. Okay, so once you have this impression about the location, what you can do is next you infer your region of interest. So for example, from this activation map, one can already infer the region of interest to be at the frontal cortex and a second region of interest at the occipital cortex. So from this, you might be then interested to extract their respective source time series. So from those voxels which have or which show strongest activation. But now the question is how to extract the source time series. So we can't do it using the transient mode, rather using the second option, which is accessing LORETA as a primary transformation. To do that, let's go to the Transformations tab. And you see here there's Spatial Signal Processing ribbon, and you see there's a LORETA transformation. So let's call the LORETA transformation. Okay, so this is the dialog of the LORETA transformation. And it enables you first to specify region of interest, which you have already inferred from the transient LORETA. So to create region of interest, the first thing to do is to create a node by clicking on this Insert Row button. So this creates a new row node. So I will create now 2 rows. One will be for the frontal source and another for the occipital source. And you can rename the node as per your desire. So I will call the first one to be frontal, and then I will rename the second one as occipital. Okay, and now the next step would be to set brain regions that should be associated to the row that you have created. So you have the possibility to group your rows either based on lobes by clicking on this Add Lobe button, where you can select the lobe of your interest. Okay, and moreover, you also have the possibility to specify whether you need only the left hemisphere or only the right hemisphere, or if you need both hemispheres of the selected lobes. And you also have the possibility to categorize your rows based on gyrus or Brodmann area by clicking on either of the 2 buttons. Okay, for now, let me click on the Add Brodmann area button. And here it enables you to select brain areas from the list of predefined anatomical region. So for our demo, I will select Brodmann area 17 for the occipital row. And for the frontal, I will select Brodmann area 10. Okay, so this way you can create a row tree. And each row tree contains 2 subnodes. So the one with a green node always tells you to which category the brain region belongs to. So in this example, the Brodmann area. Okay, and the one with a gray node always tells you the brain region from the selected category. So in this example, specifically Brodmann area 10. Okay, and detailed information about the location of a voxel within each row is also displayed in this white text box. So you can get additional information from this box. Okay, once you are done with defining your rows, you can use this Save Rows to File button. It enables you to save the tree to an XML file. So this will allow you to conveniently share your tree across different workspaces in Analyzer. And of course, it will save your time if you have to create the same row in a different workspace. Okay, and here the anatomy section over here is what I have discussed already in the slides, and it enables you to choose between various sets of standard brain templates for the display purpose. So for our demo, I will select the average 305 brain template. So this is just to introduce you to the dialogue of the LORETA transformation. But remember now, our goal is still to extract the frontal and occipital source time series from their respective voxels that showed maximum activation, which we have seen here on the right side of the transient LORETA. So I have already prepared a row tree, so let me load it by clicking on the Load Rows from File button. The file was saved with an XML format, so I will select the corresponding extension and then select the prepared row file. So I will open it. So you see, these are my 2 rows that I have created, one for the frontal source and the other one for the occipital source. So just to remind you, from the transient LORETA, we have seen that the voxel in the occipital area with the strongest activation was sitting at here -3, -74, and 29. So as you can, if you can follow me on the right side of the transient LORETA view. So this was the voxel that showed maximum activation for this occipital area. So you can also see here, it's located in the Brodmann area 7, and the lobe is occipital lobe. So I have used the information that I got from the transient LORETA view to create these 2 rows. so that the end goal would be to extract their corresponding time series. Okay, now let me show you how I created a ROI with a single voxel. To do that, first you create a node by clicking on the Insert ROI button, and then you can click in the area within the MNI brain template, so the area of your interest or the region of interest where you want to place the voxel, and then you can fine-tune the position with these yellow sliders. And once you are satisfied, you can click the button Add Nearest Voxel. Okay, what this button does is it will give you the exact location of what you have defined using the sliders if it finds a match in the source space database. But if it does not find a match, it will then provide you with the best match that is the nearest voxel to what you have selected or defined. So as you can see here, it gave me the nearest voxel that sits at -3, 17, and 22, and it's supposed to be closer to the voxel that I have wished for or defined, which is supposed to sit at 0, 11, and 90. Okay, so in this way, you can create a single voxel for your specific ROI. So for now, let me delete this node and focus only on these two ROIs. So please note that this red area, what you see in the MNI brain template, it indicates the created ROI. Okay, it does not indicate the activation of the source. The source activation is not displayed in the LORETA transformation dialog, but rather where you find it is in the LORETA transient view, which you can see here on the right side. So this means what you see in the LORETA transformation dialog is the location of the ROI that you have specified or created, whereas the transient LORETA view shows you the activation map or the spatial distribution of current density values. Okay, so this is the major difference between the two dialogs. Okay, so in addition to allowing you to create ROIs, the LORETA transformation also allows you to estimate current density within these specified ROIs for each time point. So for that, it provides you with a several output option. Let me shortly go back to the slide and briefly explain you about these output options. Okay, so within a specified ROI, you have the possibility to compute for each time point the mean of the current density vectors for only the X, the Y, or the Z components. The other option is that you have the possibility to compute for each time point the mean of the current density vectors, basically the magnitude of all the three components within a specified ROI. And the last option is that you also have the possibility to compute for each time point the power of the mean current density vectors, or basically the squared magnitude within a specified ROI. Okay, now let's go back to Analyzer and compute the current density. So for this demo, I will compute the current density or the magnitude for these two created ROIs, and by default, this option is already selected for me, so I will just press OK. Okay, now you see a node named LORETA is created as part of my history tree, and it contains new channels, which we call them as LORETA channels. So each represent the source signals corresponding to the created ROIs. So I have created a ROI that's named frontal source, and it gave me a channel named frontal source. Likewise, I have created a ROI named occipital source, and it gave me a channel named occipital source. Okay, and the output here, what you see, it looks like a flat line. And the reason behind is that the scale that I'm using right now, it fits the voltage values of a typical EEG, and it was not appropriate to display my source strength values. And basically, current source density values have indeed very small but non-zero values. So what you need to do is you have to decrease the amplitude range to be able to see the source signals. Okay, you can decrease it by using this button, scale up amplitude. Okay, let me change the view to the butterfly view mode so that you can better visualize the two source signals. So this one indicates the occipital source time series, and this highlighted signal indicates the source time series for the frontal source. So with the help of LORETA, we were able to disentangle these two sources, which was problematic at the scalp level, as we have seen it. So that means if I go to the voltage measurements at the scalp level, and if I select only those 3 electrodes, if you remember, it was hard to disentangle these two sources because what we were seeing here was the superposition of the two sources. But now, at the source level, using LORETA, the sources are clearly separated. So the advantage of source analysis, apart from disentangling the sources and apart from estimating the position and magnitude of the sources, is that it also preserves the shape of the sources. Okay, and since the shape is preserved, you can even easily tell which source is leading and which one is lagging just by looking at the time series. So you can see here clearly that the occipital source is leading the frontal source. That means the occipital source appears first and then the frontal source follows. So as I have explained earlier, the two sources were simulated in a way that there was a 90 millisecond separation or delay between them, right? So this separation is also very well preserved at the source level. And this can be checked by looking at the peak-to-peak time interval of the two sources. So let's try to check this. So I will now select the time interval. So this is the peak of the occipital source and this is the peak of the frontal source. And let's take how many milliseconds is the difference. So unfortunately I'm not hitting exactly the peak of the frontal source. Let me do that again. So I have to be careful in hitting exactly the peak. So I think now this is already the peak of the frontal source. So now let's check. It's pretty much zoomed. So let me decrease the amplitude. Okay, since I have simulated the dipoles, I already know the time point. So that's what I'm trying to figure out now. And let's try this time. Yeah, and you see, now it's exactly 90 milliseconds apart. So before, my time interval where I was indicating the peak was a bit delayed. I was not hitting exactly the peak. So that's why you were seeing 95 milliseconds. But now when I exactly hit the exact peaks of these two corresponding sources, now you can see it's 90 milliseconds. And this means the source separation is quite nicely preserved using LORETA. So, but however, the source strength is not preserved, even though I have simulated them to have the same amplitude. So as you can see here, occipital source modeled as a radial dipole has a high source strength compared to the frontal source. So which means high emphasis was given to a radial source that contributed to a maximum voltage at the scalp level, because I have simulated the occipital source to be radial. So let's check that at the voltage level. So to check which sources contribute for a high voltage values. So if I check for an electrode that is close to the simulated radial dipole, let's take electrode OZ, and it shows a high voltage value of 3.35 microvolt if you can look at the status bar of analyzer. So this was a high voltage, and let's compare it to the frontal source. Let's check for FZ, and this FZ is close to the simulated tangential source. So as you see at the status bar, the voltage is minus 0.6 microvolts. So it was less than the one at the occipital source. So this is also what is implied or visualized at the LORETA or at the source level. So this is due to the theoretical fact that radial sources are predominantly recorded using EEG. So which means at the source level, high emphasis is given to the radial source. So as you can see, occipital source has high source strength compared to the frontal source. Okay, so with the help of this simulated data, we have seen the benefits of source analysis, how helpful it was in determining the underlying sources, how powerful it was in disentangling the sources, and how reliable it is in preserving the shape of the sources. And now let's apply LORETA on a real data and see its effect. But before that, let me briefly go back to the slides and tell you about certain requirements and recommendations that needs to be taken care of before applying LORETA. So first, the prerequisites. To apply LORETA or any other source localization algorithm, you need to have the spatial arrangement of electrodes, which means you need to have either the standard or the realistic electrode positions. The other most important prerequisite that is highly specific to analyzers LORETA is that you need a time domain data. That means LORETA needs only voltage values as an input to the algorithm. Furthermore, there are few recommendations that need to be fulfilled in order to obtain an accurate source localization. It is recommended that the number of electrodes to be at least 64 or higher. The distribution of the electrodes should be uniform throughout the head. It is recommended that your EEG cap should cover uniformly the whole head. If you are using realistic electrode positions, it is highly recommended to have as much as possible low electrode misplacement error. Accurate electrode positioning can be achieved, for example, by using high precision electrode localizer like CapTrack. Last but not the least, as in any kind of scalp level analysis, for example ERP analysis, source analysis also benefits from a high signal to noise ratio, which can be achieved by having a good pre-processed data. Okay, now let me go back to Analyzer and demonstrate the experimental data. Okay, I will open Analyzer. Let's close the simulated history file. And now I will open one of the experimental data sets. So this data is a visual evoked data set. It contains 64 channels, including 4 EOG channels. And this data was sampled at 1 kilohertz. And it was online referenced to CZ electrode. For this experiment, a visual stimulus of emotional pictures was presented. And as you can see here on the navigation bar, there are 8 runs, each containing 100 such stimuli, resulting in a total of 800 stimuli. Okay, so I'm not going to demonstrate the pre-processing pipelines, but I will tell you that common pre-processing pipelines have already been prepared and applied to the data, as you can see here, like filtering, ocular correction, baseline correction, and artifact rejection has already been applied to remove physiological and non-physiological artifacts. One important point I would like to mention here is if the end goal of your processing pipeline is only to achieve the result of LORETA, then the new reference node over here is not relevant. Since the output of LORETA is reference-free, the choice of any referencing scheme has no impact on it. Okay, so after all these processing steps, the 800 segments resulting from the 800 stimuli were averaged. So this is the average data. Let me switch to the butterfly view mode for better visualization. So we have here a relatively clear representation of the P100 visual evoked potential. And now let's inspect the data using the transient LORETA. Okay, I will first select an interval around the P100, and then let's call transient LORETA. So as you can see, the source activation underlying this P100 has its maximum current density at the occipital lobe and specifically Brodmann area 17. So we can use this Brodmann area 17 as our region of interest and then try to compute its source time series using the LORETA transformation. So to call the LORETA transformation, let's go to the Transformations tab, and from the Spatial Signal Processing ribbon, let's call LORETA. Okay, to do that, I have already prepared again here a dedicated ROI tree. So let me load it by clicking on the Load ROIs from File button. And this file was saved as an XML file. So let me change to the corresponding extension. And I will select the prepared ROIs and open the file. So as you see, I have created or prepared 3 regions of interest. The first ROI is to represent a single voxel that sits in the vicinity of the strongest activation, so within this occipital lobe. So I try to create a single voxel, which is restricted by a sphere. And to do that, what I did was I placed a sphere to be centered at 4, -81, and 8. And by now, you know where I have already received this information. So this information is depicted here, if you can see at the right side of the LORETA transient view. So this is the position of the voxel that showed the maximum activation of underlying the P100 evoked potential. So I have used this information to create a single voxel. So in order to create voxels within a sphere, what you need to do is you can click in this MNI template at the place of your region of interest, and then adjust it again with these yellow sliders. And once you are satisfied, you can click then on the Add Sphere button. Okay, this is how you will create single voxels which are confined in a sphere. So it looks similar to that of creating nearest voxel. And this additional information, the radius, enables you to restrict the number of voxels that needs to be included within a sphere. So by adjusting this number, you can increase or decrease the number of voxels that should be included in inside the sphere. So if I right click and edit, and then change the radius, let's say to 8. So you see, it has increased. So the number of voxels has increased. And if I change now to a smaller number, let's say 6. So you see, I was able to hit a single voxel. So for this particular sphere, a radius of 7 millimeters or less will give me a single voxel. Okay, so this is how to create single voxel confined, single or more voxels confined inside a sphere. So if you see here on the right side of the transient LORETA, we also see additional sources that are activated on the right and left side, left hemisphere of the Brodmann area 17. So that is why I have also created 2 additional ROIs, one containing the voxels within the left hemisphere and another one containing voxels within the right hemisphere of Brodmann area 17. So R means right hemisphere and L means the left hemisphere. So now let's compute the current density or the magnitude for all these 3 ROIs. So as I've told you at the beginning, this option, the current density or the magnitude, has already been selected by default. So I only need to press OK. Okay, once it is done, it has already created a node in my history tree. And as you see, it has provided me with 3 LORETA channels, each representing the source signals corresponding to the rows that we have created. Okay, let me change the view to a butterfly view mode for better visualization. And as I said, since the scaling is very high and it only suits the EEG voltage values, I need to decrease it in order to be able to see the source time series. So now you can clearly see the 3 source time series for the 3 rows. So this highlighted source time series corresponds to those voxels in the left hemisphere of Brodmann area 17, and this middle one, or the highlighted one, corresponds to the source time series for those voxels in the right hemisphere of Brodmann area 17, and the last one corresponds to the time series for a single voxel in the Brodmann area 17, which I have placed it to be in the vicinity of the maximum activation. So as you can see here, the single voxel has a maximum magnitude as compared to these 2 other sources. And this is the reason is, since the activity is computed just for a single voxel inside a sphere, you get a high source strength. Whereas if you see these 2 source time series, the source strength were created by averaging all the voxels which were confined in the left hemisphere, and for this one, all voxels were averaged. The source strength of all the voxels that were confined in the right hemisphere were averaged. So due to this reason, you see a smaller strength as compared to the single voxel. So once again here, the shape of the source time series is preserved, and it has the same shape as that of the P100 evoked potential. So this result is now on a single subject level. Now, let me demonstrate you how you can perform group level source analysis across subject. So as you see here, I have additional 3 subjects, and I have applied the same pipeline for all the subjects. Okay, so in order to do group level analysis, what we need to do is first to perform grand average. Okay, so let's go to the segment analysis functions ribbon. Then here we have the result evaluation. If we open that, we will get the grand average module. So here we need to compute the grand average from all average nodes which contain the averaged evoked potential across all subjects. Okay, so I will give it a name. So this is our grand average containing the average of all evoked potentials across all subjects. So across subject 7, across subject 8, subject 11, and subject 14. Okay, and now I will include all these subjects and compute the grand average. Okay, since grand average is a secondary transformation, I will go to the secondary tab. So this is our grand average data. And now let's apply LORETA on this grand averaged data set. So we go to the spatial signal processing ribbon and call LORETA. And let's use the same ROI tree which we have used for the single subject. Okay, and let's also compute the current density or the magnitude. So as we have done similarly for the single subject. Okay, I will press OK. Okay, so in a similar way, we now get the source time series for each of the 3 rows. In order to have an impression about the result of LORETA on the grand average level, as an example, let me select the source time series of a single voxel, which I have placed within the vicinity of the maximum activation. And let me decrease the amplitude so that you can see it. And now in our next step, what we want to do is to overlay it, and so that you can compare and contrast how it differs to that of the single subject level. Okay, so for that, in order to differentiate, let me change the line width of the source time series by going to the settings of grid view and select the overlay tab. So I want to make it bold, so I will change it. I will change the line width, and let me change the color so that it will be clearly visualized for you. Okay, I will say let the reference node have or show me the channel name, which means the BA17 for a single voxel, and then I want the overlays to have the name of their history file. So I will put here H. Okay, now it's done. So now I will go to the primary tab and I will try to overlay the LORETA or the time series for each subject. Okay, so you see this one is for subject 7. I will keep on overlaying. So this one for subject 8, this one from subject 11, and this one from subject 14. Okay, so we have overlaid all our LORETA results, and as you see here, the bold line is the time series from the grand average, and it nicely lies in between the single subject source time series. Okay, so you can apply this nice feature of overlay to compare and contrast results between subjects or between conditions. Okay, so with this, I will end now the demo session. So let me briefly go back to the slides and then summarize this section. So I have showed you the different ways of accessing LORETA in Analyzer. The first option was the transient mode. It is used for inspection purpose, resulting in a temporary display of a source activation map. The source estimation is done only for the selected time interval and displays the magnitude of the current density vectors within the time interval associated to a specific voxel. The other mode of operation is as a primary transformation. This option enables you to create or load your rows from a predefined source space. For the created or loaded rows, it then provides you with the option to compute for each time point the mean of the current density vectors for each component separately, or the magnitude of all the 3 components, or the squared magnitude. So we have the source activation map and its time series. What should we do with the source time series? For example, you might be interested of extracting some information or measures for further statistical analysis. Without going in further detail, I will briefly point out the measures that can be extracted from the current density values and the ways to extract them. So if you intend to export the output values for each time point, you can use the generic data export module in the same way as you might have already used it to export other data. And if you intend to export the area information or the mean activity around the strongest source signal, you can use the area information export. You might be interested to detect the peaks of the strongest source signals. So to do that, you can apply the peak detection transformation. And to export the values corresponding to the detected peak, you can use the peak information export module. Okay, so far we have seen and discussed interesting capabilities of LORETA in source imaging. It is also worth mentioning its limitations to be more cautious in interpreting the results. A general limitation is that it has a low spatial resolution. This is due to the assumption that the activity of neighboring neurons over a relatively large distance is correlated. As a consequence of this assumption, LORETA provides blurred activation maps, for example, activations that include homogeneous areas in both hemispheres or 2 distinct areas along a sulcus will be over-smoothed. The other limitation, which is specific to Analyzer, is that frequency domain and time frequency domain source analysis are not supported. This leads me to inform you the do's and don'ts of LORETA in Analyzer. The only valid input is a voltage value, that is, a time domain EEG data. The outputs from frequency domain and time frequency domain data analysis, or the outputs from any statistical analysis, shall not be used as an input to LORETA. So please be aware not to apply LORETA on these transformations, as it violates the mathematical and physics underlying all assumptions. Further details on the topic of misusing LORETA can be found in this link. Okay, before I provide you with the take-home message, I want to address some of the questions of interest that we received during the live webinar. Okay, so the first question was, if I want to inspect the differences in the source activation map across conditions, is there an overlay feature for the activation maps? So the answer is, overlaying source activation map is not possible because no node is created to store the results. However, there is a workaround in Analyzer that enables you to visualize the similarities or differences in the source activation map across conditions or across subjects. So to demonstrate this, let me go to Analyzer. So let's say you want to compare activation map of the average node across each subject, okay? And this is achieved by making use of the additional LORETA feature, which is an add-in. So you can access it by going to the add-ins tab here, and you will find here the LORETA add-in. So if you click on it, it will generate an extra or additional window, and it enables you to easily switch between conditions or between subjects. As you see, if I click on different nodes or different conditions, it will allow me to compute the current density on the fly over the entire time interval. Okay, so you don't have to close the dialog. You can easily compute the current density values for that specific node. So this way you can compare source activation between conditions or between subjects on the fly. So this is a nice feature. And now moving on to the second question, how many trials are necessary for a valid source localization? Isn't 800, as in your example, a really large number? Okay, so theoretically, the more segments you have before averaging, the better is the chance to get an increase in signal-to-noise ratio. Therefore, it's good to have as much segments as possible. But so far, I'm not aware of a golden rule pointing to a certain amount of trials to achieve reasonable signal-to-noise ratio for a valid source localization. So at this point, I would like to suggest you to check for literatures related to your research area. Okay, and now I will go on to the third question. It says, after computing the maximal voxel, how can I determine the localization accuracy? So if you have an a priori information about where the strongest activity should occur, so maybe from some peer studies or from literatures, you can estimate how well or how fairly LORETA has localized the strongest source using the technique called Euclidean distance measure. So I can show you this using the simulated data. So let me open the simulated data set. So from our transient LORETA, we have seen that the voxel with the strongest activation was sitting at -3, -74, and 29. So this is what the algorithm provided me. But I know a priori when I was simulating the dipole that it was sitting at -3, -74, and 1. So you see, there is a difference in the z direction. So I modeled it to be sitting at 1, and the algorithm provided me with 29. So if you subtract now 29 minus 1, you will get 28 millimeter. And as I have already discussed, the size of a single voxel is 7 millimeter. So you can easily say that it is 4 voxels apart from the true source. So in this way, you can infer the precision in the localization judgments. Okay, and now moving on to the fourth question. Should I calculate LORETA on individual averages and then perform the grand average of LORETA, or should I calculate LORETA on grand average data? So from the signal quality point of view, if you apply LORETA on grand average data, you will gain a better signal to noise ratio. But on the other hand, if you apply LORETA on individual averages and then compute the grand average of LORETA, sources might be biased by the presence of noise in each individual average. But in general, it depends also on your research question. Do you intend to do subject-level analysis or group-level analysis? Subject-specific source analysis will benefit if you have individual electrode positions and individual MRI scans. LORETA's implementation in Analyzer, as we have learned so far, uses a standard brain template. So due to this reason, subject-specific analysis is not yet feasible in Analyzer. However, you can apply it for group-level analysis by applying it on the grand average data. So this way, you can inspect typical source variations within the study cohort. Okay, now I will move on to the final question. How is the source localization affected by removing noisy channels in advance, thereby also amounting to a smaller number of electrodes, which is less than 64? Okay, so removing noisy channels not only decreases the number of electrodes, but it might also create non-uniform distribution of electrodes throughout the head. So this will then result in low localization accuracy because source localization is quite sensitive to the number of electrodes as well as the spatial distribution of electrodes. This is what also we have discussed in our recommendations. So therefore, it is advisable to make use of artifact correction techniques while preserving a uniform montage. So I hope that answers the question, but for further detailed explanation, I would suggest to have a look at the paper by Christophe Michel and his colleagues, which I will provide you at the end of the webinar. This paper shows the relationship between the number of electrodes and localization accuracy, as well as discusses on how to handle artifact contaminated channels. Okay, so with this, I will end the question and answer session. I hope you have benefited from these compiled questions. So let me now go back to the slides and give you some concluding remarks to recap what we have been discussing in today's webinar. So the important take-home messages are the following. The spatiotemporal EEG dynamics partially encode the information about the underlying neural dynamics. LORETA provides a reasonable source configuration, although it has its own limitations. Please be aware of the prerequisites and misuses of the LORETA transformation. Region of interest for LORETA analysis should preferably be defined based on previous scientific papers related to your research area. This can be incorporated together with the exploratory analysis using the LORETA transformation, either from the transient view or via the add-in option. In general, although source analysis is a difficult problem, it is really a great technique devoted to modeling and estimating the spatiotemporal dynamics of neuronal current sources throughout the brain. However, there is no gold standard method available yet, so be cautious when interpreting the results related to the location and magnitude of sources. For further readings, we have some recommendations where you can get quite a nice introduction to LORETA, which was originally developed and described by Pasquale Marchi on his 1994 and 1999 articles. The implementation of LORETA in Analyzer is also based on these two references. In his other article published in the same year, you can also find a nice comparison of the inverse methods, where he compared the different source localization algorithms from the family of non-parametric approaches. Another nice article comparing and discussing all families of inverse methods can also be found from the paper of Christophe Michel, as I've mentioned in our question and answer session. So from our side, we also offer materials, and one is of course via our Analyzer user manual, where you can find detailed information on the two modes of LORETA operation, LORETA as a primary transformation and the transient LORETA transformation in their dedicated chapters. We also have 2 press release articles in our website. The first one, which is entitled Beyond Surface EEG, an Invitation to Source Analysis with LORETA, will give you a quick grasp of LORETA in Analyzer. The second one, as the name implies, it provides with a step-by-step explanation on how to make the best use of LORETA as part of your processing pipeline. The article provides you with solutions if you, for example, have questions like, how can I create or load rows containing voxels of my interest? What is the effect of combining different regions of the brain in a single row? And many more tips. So I would say it is definitely worth reading it. And if you have further questions related to this webinar or any other analyzer-related questions, please don't hesitate to contact us via support@brainproducts.com. So with that, I would like to thank you all for watching our webinar. On behalf of the Brain Products family, I wish you all good luck with your further analysis and enjoy localizing your EEG sources. Have a good time. Bye-bye.